Welcome to the fascinating world of computational biology. Imagine you are walking into the largest library on Earth, but instead of neatly organized books, the shelves are filled with billions of loose pages containing random letters. To make sense of this massive ocean of biological information, we use specialized digital filing systems called biological databases. Today, we will explore what these databases are, how they are categorized, and the mathematics behind their explosive growth. Now that we understand the scale of the problem, let us look at why these systems are so essential. In biology, we face a unique challenge because modern technology allows us to read the DNA of living organisms at an incredibly fast pace. If you wanted to find a specific genetic recipe, it would be completely impossible without a proper cataloging system. Biological databases are the beating heart of modern biology. They act as highly organized, searchable computer archives that store biological data, such as DNA sequences, protein structures, and metabolic pathways, allowing scientists worldwide to collaborate. To manage this vast information, we divide these databases into three main types, starting with the foundation. First, we have primary databases. You can think of these as raw archives, where scientists directly deposit their experimental results. It is very much like dumping raw ingredients into a pantry before you start cooking. When a researcher discovers a new gene, they first submit the raw sequence to a primary database, like GenBank, to claim their discovery and share it with the world. This raw data is the starting point for all subsequent biological discoveries. Once the raw data is stored, it needs to be processed, which brings us to our next category. Second, we have secondary databases. These contain highly curated, analyzed, and annotated information that is derived directly from the primary databases. If primary databases are the raw ingredients, secondary databases act more like a refined recipe book. Other experts will analyze a raw gene, figure out exactly what protein it makes, and store that refined knowledge in a secondary database, like SwissProt. This structured ecosystem prevents computational biologists from wasting years trying to find reliable data. With both raw and refined data available, scientists need a way to navigate it all seamlessly. Finally, we have composite databases. These are unique because they do not store new data themselves. Instead, they act as a massive search engine that links primary and secondary databases together. This allows you to search across multiple platforms at once with a single query. Without this overarching search capability, bringing together information for designing new life-saving drugs or tracking viral outbreaks would bring medical research and genetic engineering to a complete halt. Before we move on to the mathematics of how these databases grow, let us visualize their relationship. Here, we see a clear flowchart illustrating the flow of biological data. At the top, we have the primary databases, representing the raw DNA data entering the system. Following the arrows downward, this raw data is processed into the secondary databases, where it becomes curated and annotated. Notice the arrows pointing from both the primary and secondary databases down into the composite databases at the bottom. This shows how the composite search interface pulls information from all levels, creating a unified, easily searchable network for researchers. Now that we understand how databases are structured, we must address a massive logistical challenge. One of the most incredible things about biological databases, especially primary ones like GenBank, is how rapidly they are expanding. Because DNA sequencing technology is becoming cheaper and faster every day, the amount of data we store does not just grow by a fixed amount each year, it grows exponentially. To understand and predict how much computer storage and processing power we will need in the future, computational biologists rely on the mathematical model of exponential growth. This brings us to the mathematical equation that models this rapid expansion. This formula comes from the natural laws of continuous growth. Let us break down every symbol. N of t represents the total number of biological sequences in the database at a specific future time t. And 0 stands for the initial number of sequences, present right now. The letter E is Euler's number, approximately 2.718, the fundamental base for continuous growth. The variable r represents the continuous growth rate, expressed as a decimal. If the growth rate r increases, the total number of sequences will explode upward much more rapidly, requiring massive new supercomputers. Let us put this formula into practice with a realistic scenario. Suppose a newly established primary database currently holds exactly 100,000 sequences and is growing at a continuous rate of 20% per year. We want to calculate how many sequences it will contain after 5 years. We chose the exponential growth formula because the growth is continuous. First, we substitute our starting amount, our decimal growth rate of 0.20, and our time of 5 years. Multiplying the exponent gives us exactly 1. Raising e to the power of 1 leaves it unchanged, so we multiply 100,000 by 2.71828. Our final answer means the database will grow to approximately 271,828 sequences, illustrating the rapid expansion biologists must manage. Having seen how to calculate future size, we often need to answer a more specific question for capacity planning. A very common question in computational biology is how long it will take for a database to become twice as large as it is today. This specific time period is known as the doubling time. If a protein database is growing rapidly, knowing exactly when it will double is a critical calculation for planning future server upgrades. Interestingly, as we will soon see mathematically, the time it takes for a database to double depends exclusively on its growth rate, regardless of whether it currently holds 10 sequences or 10 million sequences. To make these capacity predictions easier, we use a specialized version of our growth equation. This formula is derived directly from the exponential growth model by setting the future amount to twice the initial amount. In this formula, t is the doubling time we want to find. The numerator is the natural logarithm of 2, which is a mathematical constant, approximately equal to 0.693. The denominator, r, is our continuous growth rate, expressed as a decimal. Notice that the initial size of the database is completely absent from this formula. If the growth rate, r, increases, the denominator gets larger, which physically means the time required to double becomes much shorter. Before we prove where this elegant formula comes from, let us see it in action. We're given a protein database growing at a continuous rate of 35% per year, and we're asked to find how many years it will take to double. We use the doubling time formula because it directly relates growth rate to the time needed to double. We plug in 0.693 for the natural log of 2 and 0.35 for our decimal growth rate. Dividing these gives us approximately 1.98. Physically, this final answer means that at a 35% growth rate, the database will double its server storage requirements in just under 2 years. Now that we have used this formula, Let us understand where it comes from by deriving it step by step. This first chunk of the proof is setting out to establish the mathematical condition for doubling. We start with our standard exponential growth equation. Because we want to find the exact moment in time when the database has grown to double its original size, we replace the future amount variable with 2 times the initial amount. Notice that the initial amount variable appears on both sides. By dividing both sides by the initial amount, we completely eliminate the starting size from our calculation. This proves mathematically that doubling time is independent of the current database size. Next, we will solve for time. So, we have proven that the initial size cancels out, leaving us with 2 equals e to the power of rt. This brings us to the final steps of our derivation, where we must extract our variables from the exponent. To do this, we apply the natural logarithm to both sides of the equation. The natural logarithm is the mathematical inverse of the exponential function, meaning it effectively cancels out the base constant e. This algebraic trick brings the growth rate and time variables down to the main line. Finally, we rearrange the equation to isolate the time variable by dividing both sides by the continuous growth rate. This gives us the standard, universally used formula for calculating doubling time. We have covered a lot of ground today, so let us review the most important ideas. First, remember that biological databases are essential digital libraries. Primary databases store the raw, unpolished experimental data, while secondary databases provide highly curated and analyzed insights. Composite databases link everything together for easy searching. Second, because DNA sequencing is so fast, these databases grow at an exponential rate, which we can model mathematically to predict future sizes. Finally, the time it takes for a database to double depends entirely on its growth rate. By mastering these concepts, you have taken your first major step toward understanding how we organize the very code of life. Great job!